Proper cryopreservation of mammalian cell lines ensures long-term storage and high post-thaw viability. In this tutorial, we will demonstrate step-by-step how to correctly freeze both adherent and suspension cells. Start by ensuring that adherent cells have reached between 80 and 90% confluency to ensure they are in optimal health before freezing. Healthy cells improve post-thaw recovery and experience less stress during the freezing process. Begin by aspirating the spent medium carefully to avoid disturbing the cell monolayer. Rinse the flask with phosphate-buffered saline solution to remove any residual serum, as it could inhibit trypsin activity. Aspirate the phosphate-buffered saline solution, then add trypsin EDTA solution to detach the cells. Make sure the cells are evenly coated with the solution. Incubate at 37 degrees Celsius for 1 to 3 minutes, or until the cells round up and detach. Once detached, neutralize the trypsin by adding complete growth medium. Transfer the cell suspension to a centrifuge tube. For suspension cells, harvesting is simpler. Collect the cells by transferring the culture medium directly into a centrifuge tube. Before proceeding with cryopreservation, count the cells to determine their concentration. This ensures accurate cell seating in each cryogenic vial and maintains consistency across experiments. Centrifuge the cells for a few minutes to pellet them. After centrifugation, remove the supernatant. Gently resuspend the pellet in an appropriate freezing medium. Distribute the cell suspension into pre-labeled cryogenic vials, ensuring that each contains a consistent number of cells for reproducibility. Label each vial with essential details such as cell type, passage number, and date to facilitate future identification. To minimize ice crystal formation, use a controlled cooling rate of approximately -1 degree Celsius per minute. Place the vials in a freezing container and store them at -80 degrees Celsius overnight. After 24 hours, transfer the cryogenic vials to a liquid nitrogen tank for long-term storage at -196 degrees Celsius. Maintaining proper storage conditions is essential to maximize cell viability upon thawing. Now, let's dive into the theoretical principles underlying mammalian cell cryopreservation. Cryopreservation enables long-term storage while maintaining cell viability, genetic stability, and functionality. However, improper freezing techniques can cause damage and lead to loss of function. During freezing, ice crystal formation can disrupt cell membranes. Cryoprotectants like dimethyl sulfoxide help reduce ice formation by stabilizing membranes and preventing osmotic stress. A slow freezing rate of -1 degree Celsius per minute allows water to exit the cells gradually, preventing intracellular ice formation. After initial freezing at -80 degrees Celsius, cells should be transferred to liquid nitrogen storage to maintain long-term viability. Selecting the appropriate cryopreservation medium is essential for optimal post-thaw recovery and viability. The choice of freezing medium depends on the cell type, as different cells have specific requirements for optimal cryopreservation. Cryopreservation media generally consist of a base medium, a cryoprotectant, and a protein source. The cryoprotectant and protein help minimize cell damage during freezing and thawing. Serum-containing media provide additional protection by supplying essential proteins and growth factors that enhance cell viability. Common formulations include complete medium with 10% glycerol, complete medium with 10% dimethyl sulfoxide, 50% cell-conditioned medium, and 50% fresh medium with 10% glycerol or 10% dimethyl sulfoxide. Serum-free formulations are suitable for cell lines adapted to serum-free conditions or applications requiring the absence of serum components. Examples include 50% cell-conditioned serum-free medium and 50% fresh serum-free medium with 7.5% dimethyl sulfoxide, fresh serum-free medium with 7.5% dimethyl sulfoxide, and 10% cell culture-grade bovine serum albumin. To ensure proper cell freezing, calculating the required cell suspension volume is essential. Consider the following scenario. A researcher wants to freeze 10 cryogenic vials of adherent cells at a concentration of 1 million cells per milliliter, using 1 milliliter of freezing medium per vial. This means a total of 10 million cells are required. After counting, the researcher finds that the cell suspension has a concentration of 5 million cells per milliliter. To obtain 10 million cells, the required volume is calculated as follows. Thus, the researcher needs to take 2 milliliters of the cell suspension and mix it with 8 milliliters of freezing medium. Now that we have covered the theoretical principles, let's review common mistakes to avoid. Using an inappropriate cryoprotectant can lead to poor recovery. Always select an optimized freezing medium based on your cell type. Rapid freezing or inconsistent temperature control can cause cell damage. Always use a controlled rate freezing process to maximize viability. Excessive dimethyl sulfoxide is toxic, while too little fails to protect cells. Keep it between 5 and 10% for a balance between protection and cell health.